Thank you. Thank you very much, Mindray, for organizing SFCC. And on behalf of IFCC Executive Committee member of Scientific Division, I congratulate Mindray for organizing this academic treat and being a Diagnostic Division member of CAHO and also a member of ACCLNP. It's my privilege to have this talk and in front of this great audience, and I'm really sorry that I can't, because there is some institutional ethics committee things are going on, so because of that I have to attend that. So today my talk is on proper implementation of IPOQ-PQ, the tools to ensure the quality of results. So for us diagnosticians, every blood sample tells us a story. Since morning we heard Dr. Kupil Venkatesh sir, then Dr. Manisha, and then Dr. Aparna, and now Dr. Prajjal. So we have seen how different, how quality control tools, as well as how different case studies, how being alert, and how implementation of the protocols and harmonizing with the clinicians, how it is important. So let's see, this is my acknowledgement to all the diagnosticians present here, biochemists, pathologists, microbiologists present here, to acknowledge all of our efforts. Maybe we are seeing slides under microscope, or many of us are doing tests in autoanalyzer and interpreting this RT-PCR. So we are not behind curtain, and also the stakeholders like IBD, stakeholders like Mindray. So everybody, we have seen during COVID time how all stakeholders played a very significant role. So this is my painting to acknowledge all of our efforts. So the genesis of my today's talk is this TSH report. So this, like Dr. Aparna mentioned during her talk about AMAs, so many times, like this TSH is a very simple test we are doing routinely. And many times we see patient goes to 3 different labs during the short span, or maybe same day they send 2 samples to 2 different labs, and seeing also morning and evening the difference in values. So like one lab reported 0.02 microinternational unit per ml, another reported 0.67, and we reported 0.1. So then when 3 reports same time, the patient goes to the endocrinologist, then endocrinologist is thinking what lab result to believe, and which is a good lab. And so this is us, our lab system. If we see this, we are coming with burden, patients are coming with burden of disease, then we implement our intelligence, discipline, and creativity. And then patients are going with reports, and sometimes we get called, like the previous talk, he mentioned that when he was having the breakfast, he got the call, and luckily the hematologist was with him. So we get the call from clinicians, that Barnali, what creatinine value you reported, what or many times maybe they want to do, like just now I received a call that they want to do IPTH in tip, the needle tip. So how to report that? See, IPT in needle, saline. So sometimes we need to guide also the clinicians or radiologists that you have to do saline wash for PTH, and then send the saline wash PTH sample for cystic fluid. So these are our role. So sometimes they may be happy, our clinicians, or sometimes our patients are complaining, oh, why there are 3 different TSH report. So because of that, it is our role, this is our today's lab system. And if you can realize this can be you or me, being an ABL assessor or CAP inspector. So we go for sample audit for College of American Pathologist 1 day or for NABL 2 days. And based on our observation, we say, oh, this lab seems to be very good. There is no non-compliance, or there are so many non-compliances or deficiencies. So this is our today's lab system. So this is the genesis of like, why do you think that there are three different reports at the same time? Do you think that sample transport or sample integrity is issue? Because being an assessor, many times I have seen all the transport media for sample transport, not seen yet the elephant, but hopefully I'll see that too. But so do you think the sample integrity is an issue, or do you think the analytical variation between instrument to instrument is real challenge? So for that, I will take the reference as thyroid hormone standardization, because I was part of IFCC Committee for Standardization of Thyroid Function Test, and also being in the scientific division, we are trying to standardize and harmonize many of the immunoassays and biochemistry assays. So are we nearly there yet for thyroid hormone standardization? So for any like biochemistry or immunoassay testing, so can IQ, OQPQ really help us? So I can see Dr. Tina Liu from Mindray. So last time when we met, I think in Rome, so we discussed that at like, you know, at length, like how all the IVD, because we see a lot of variation between instrument to instrument in the harmonization and standardization. So how we can harmonize the test in biochemistry and immunoassay platform? So these are some of our papers, published papers in European Thyroid Journal, Clinica Chimica Acta, and Clinical Chemistry on thyroid hormone, like how to harmonize and standardize. So now these are based, some are based on reference intervals, some are based on like recalibrations and harmonizing, or based on the APTM, or purpose mean how we can establish after recalibration. So these are some of the papers. And now the outline of my today's talk is understanding IQ, OQPQ or for IVD devices, then installation qualification, what are the tangible and intangible aspects related to installations, then requirement of the IQ, and then operational qualification and performance qualification. I remember when I started my journey in lab, so we initially we were only bothered about the performance qualification in lab. About IQ, about OQ, we used to think whoever the manufacturer or whoever the IVD person supplying us the instrument or kit, so they will do the IQ, OQ, PQ. So why I should bother? I should only be bothered about the part of PQ or part of the performance qualification. But that's not correct. So now, from IQ, from installation qualification to operational qualification and performance qualification, the lab, as a lab personnel, we need to validate that, verify that, and we need to sign that. So let's see how to do that. So IQ, OQ, PQ protocols are methods for demonstrating that equipment being used or installed will offer a high degree of quality assurance. The goal is the process validation to deliver a stable IVD that offers consistent performance. Variation should be minimal and predictable, and the process validation plan will provide the framework. Like we can start, if it is a new lab, we can start from design qualification, then we can shift to the installation qualification. When we talk about installation qualification, it should answer, is everything installed correctly? Operational qualification, is everything operating correctly? Are operating limits understood? Then performance qualification, did it produce the correct result? Is the process stable and capable? So this is that full chart where from initially when we do this process, we form the validation team, then we create our validation master plan, then we create our protocols, perform the design qualification, then we perform IQ, OQ, PQ, and prepare the final report, and then we validate it. So IQ is, it verifies the instrument being qualified and have been installed and configured according to manufacturer's specification, and it is documented collection of activities necessary to establish that an instrument is delivered and designed and as designed and specified, and it is properly installed in manufacturer specified environment. So what should be the checklist? Then for checklist, the location of installation and necessary floor space, documentation, any and all computer control instrumentation, gathering all manuals and certifications, and then properly unpacking and cross-checking the instrument, examining instrument and components for damage, ensuring correct power supply, documenting firmware versions and serial numbers, environmental and operating conditions, checking software system installation and accessibility, recording calibration and validation dates of tools used for IQ, and verifying connection and communication with the peripheral units. So IQ verifies that the instrument being qualified as well as subsystem and ancillary systems have been installed and configured according to the manufacturer specification or installation checklist. The FDA's IQ definition offers a useful statement of the overall goal, documenting that the system has the necessary prerequisite condition to function as expected. So this comes along with the current good manufacturing practice CGMP requirements. So if we want that checklist, so components and equipment, software and standard operating procedure, then calibration and testing, then installation, software and system testings, records, electrical records, and verifying the electrical things in the lab, then signals and alarms, revalidation, facility condition, and documentation. So now IQ, there should be an SOP, checklist, or some other documented process that defines the standard installation procedure for each type of system or deliverable being installed. So these requirements may come from design specifications, system specifications, manufacturer's recommendation, and data sheet. So IQ verifies and documents key aspects of the installation meet approved requirements. So this, if we see the checklist, so this is one checklist from our lab. So these are the checklists like operator manuals, environmental parameters, instrument delivered undamaged, transport locking, all connection correctly installed, instrument positions according to installation manual, instrument boot process successfully, and then specification met or not, mechanical adjustment, auxiliary components position or not. So these are different components where we should, when we are signing IQ report, we should see that that are installed properly or not. Or if there is a deviation, what corrective action, what preventing action the IVD has taken, and if the deviation resolved satisfactorily or not. Now coming to the OQ. OQ's purpose is to determine that the equipment performance is consistent with the user requirement specification within the manufacturer specified operating ranges. So after we have installed IQ, then comes the next step is OQ. This means identifying and inspecting equipment features that can impact final quality. So during OQ, all items in the test plan are tested and their performance is thoroughly documented. So OQ is associated with equipment performance to ensure that the function of the machine, measuring devices, utilities, and manufacturer areas perform as intended throughout all anticipated operating ranges in the selected environment. So actually, after completion of OQ, we permit a formal release of the performance qualification process. So what should be the components of OQ? Internal hardware calibration, software verifications, maintenance verifications, parameter calibrations, internal quality control, precision of analysis, verification of all the above with respective documents, documentations of the above step. So this is like the operational qualifications or OQ. These are the like calibration, calibrated successfully or not, then OQ, quality control, then accuracy check successfully or not. So this is the validation process. And then this is the checklist and where we sign the OQ results acceptable or not. So the final steps where we as a diagnostician play a very, very significant role. The final step of qualifying equipment is the PQ. So in this phase, the qualification and validation team verifies and documents that the equipment is working with the reproducible results within a specified working range in laboratory setting. So team must create a detailed test plan based on the process description. So we call sometimes as a process performance qualification or performance qualification. So the manufacturing conditions such as equipment limits, operating parameters, component inputs. So this all should be mentioned in PQ. So like in manufacturing condition, equipment limits, operating parameters, component inputs, and then calibrations, a thorough list of data that should be recorded or analyzed during test, calibration, and validation. So like the protocols can be precision, linearity, sensitivity, accuracy, method comparison, carryover study, reference interval. So I will give some of the process we have done in our lab. So yes, our lab life is these philosophical lines of Albert Einstein. Life is like riding bicycle. To keep your balance, you must keep moving. So in our lab life, we have these two wheels of the bicycle. One is the wheel of quality. Second is the wheel of cost. Wheel of quality depends on accuracy, precision, reference interval, and wheel of cost depends on man, machine, and material, three magic M's. So we need who will do the balance between these two wheels to have smooth ride in our lab life. So this balance will be done by harmonization and standardization and proper IQ and OQ and PQ. So if we do proper IQ and OQ and PQ, so in that the variation and between methods, it actually we can harmonize our protocols quite a bit. So verification and validation play a very significant role, as we know ISO 15189 2022, and also the CAP checklist mentions that verification and validations is very important. So how we should verify any, so when it comes to we start any new test or any new instrument, so the verification protocols should consist minimally this like analytical accuracy, run samples by both new method and comparative or gold standard methods to evaluate the bias, then verify the precision, then checking the reference interval according to CLSI EP28A3C. So we check the reference interval and verify, I'm in CLSI standardization committee. So there are many difference in opinion, like I have seen, like also I'm part of ICMR reference interval advisory board. So when we talk about direct reference interval and we compare that with the indirect reference interval, so there are between our diagnosticians, still there are many variation in opinions how to establish the reference interval. Should we go for direct method or should we go for indirect method? So because of that, when we are doing verifications, it's very important to verify the reference interval. Now verify the LOB and LOD, limit of detection and limit of blanks, by running 20 blanks or zero calibrator or low patient samples, we can verify that. Then linearity verification. I remember during my CAP inspection, I have done everything, but I forgot, like I thought we are taking linearity surveys in CAP. So why we need to do, before releasing any patient samples, why we need to do linearity verification. But I got deficiency because of that linearity verification. Range that can be reported without dilution or pre-treatment of samples. So as we call that as analytical measurement range, or AMR. So AMR is without dilution or pre-treatment of the samples. And CRR, or clinically reportable range, or extended measuring interval, according to CLSI EP 34. So now the term CRR is, we call it as the extended measuring interval, or EMI. So range that can be reported with dilution or pre-treatment of samples. When we talk about immunoassay, or like Dr. Aparna told about us, different fertility assays, LH, FSH. So we see, like, you know, the IVF specialist many times gives us a call that, Barnali, why you are not reporting the exact value? Why you are giving more than so and so in while reporting? Or sometimes the tumor markers, you will get call from oncologist, why you are not reporting the exact value? Why you are reporting more than so and so? So I need exact value because I need to monitor the patient. So many times it's very difficult for us diagnosticians to give the reportable value because of this extended measuring interval, or EMI. Why? Because instrument manufacturer, IVD manufacturer, like Mindray, so they do their validation, and in that they mention that you can dilute the sample till that range. Beyond that, we are not supposed to dilute. So because of that, we can't dilute any samples till infinity. So whatever the manufacturer mentions, whatever the limits manufacturer mentions, we can verify that, and we should have our CRR or EMI chart displayed in lab, so that no technical staff, even at night, release any report which is beyond extended measuring interval, or CRR. So this is how much time I have. Okay, so this is how we actually adopt the reference limits. So these are manufacturer suggested. I'm audible, no?
You have, no, I think time is done, Barnali.
Time is done. So then I will not, I have already said this. So this is, I'm just like, you know, how we establish the reference interval. So we, in 20 sample, we do verification, we can do, but suppose we want to establish, then we can do in the CLSI say 120 if it is following Gaussian distribution, but if it is not following Gaussian distribution, then 200 samples. So these are some of the, like, how we should check the accuracy. So this is our accuracy check. Then how we should do the precision, like 5 replicates for 5 days, 25 replicates in all. So yes, data, and data. I can't make bricks without clay. So like in biochemistry lab or immunoassay lab, so when we need to establish our IQ, QPQ, when we need to establish our verification protocol, we have to actually go a lot of statistical analysis, and we have to give importance to our data. Thank you.
Thank you, Dr. Barnali. For you, unfortunately. Any questions for Dr. Barnali? You, just a second. Huh?
Hi, Dr. Barnali. This is Dr. Dhanlakshmi from Apollo Chennai. On the operational qualification of the instrument, what is your thought on including the environmental aspects of using the new instrument in our laboratory?
Yes, ma'am. We should actually
Hello, ma'am. Good afternoon.
Good afternoon.
I am Dr. Manoj from Ghaziabad, Uttar Pradesh. My question related to POCT analyzer.
Yes.
POCT analyzers, the manufacturers not provide OQPQ IQ for the POCT analyzers. So what to do in this case?
So, Dr. Puninai, for that we need to have the IQOQ, proper IQOQ PQ, because that needs IQOQ PQ. So if they are not providing, then this is our role, like, let them provide the IQOQ and we can do the PQ on our own.
Okay. Thank you. Thank you.
Okay, I think, yeah.
Great presentation, Dr. Barnali. So regarding the dilution of recommended by the manufacturer, in certain parameter we have to extend the dilution beyond the manufacturer recommendations. Then what would be the protocol and how to validate that dilution?
So we can give in written report the dilution, because I know a couple of the legal case where the lab has reported, like, beyond the limit of the manufacturer, then nobody can, like, because they will ask another expert opinion, nobody can support that lab. So because of that, many times when we get the call, and mainly for the reference lab, because the second reference lab may be giving the diluted exact value for the, who can verbally inform that this is the value we have got, but because of the manufacturer recommendation we can't go beyond that. So better to stick to like extended measuring interval or CRR in our report, and verbally we can inform that this is the report, and if you ask, then we can do the dilution. So this protocol we actually, well, in my practice I follow.
Thank you.
But written report is better not to give, because I have seen many 2-3 cases where in written report the dilution given beyond CRR or beyond the EMI, so then there is difficulty to give the expert opinion.
I think
I hope
Dr.
I'm
Barnali,
correct.
yeah, I think very well said. We do face this problem in, particularly in commonest thing like TSH. Oh, you give more than 100, tell me how much it is. Okay, so I think that's the, I'm completely convinced it's better to stick to the.
Actually for TSH it's not much, if I give 2000 or if I give more than 100, so that endocrinologist will do the same treatment.
Exactly.
Yes, but tumor marker, I have faced the many reference lab many times say we can't follow this because of the tumor marker, or like you have mentioned during IVF specialists.
Yeah.
Many times they will not accept that if you are not giving the exact
value. So it's better, I think what very, very well said, it's better to give the printed report with those instructions of
Extended
manual,
yes.
and you can say, okay, verbally, if you're going out of your way for the clinician, because legal angle is also very, very important, and very important point for point of care. Because that is what is coming up and that's what is the future. In case they are not giving us, it's I think the onus is on us to get the PQs done well. Thank you, Barnali. Superb presentation, and we miss you here.
Thank you. I am missing. Really missing.
So thank you so much. Thanks.
Thank you.
So I think Shama will show you the certificate online.
Thank you. Thank you, Dr. Barnali. It was pleasure hearing your talk. So, Dr. Barnali,